Sorry, we'll have to figure that out. Hopefully I can actually get it. Yeah. Okay, so building blocks. Your building blocks are composed of an alpha amino acid. This is going to consist of the alpha carbon linked to an amino group, a carboxylic acid group, and a hydrogen atom, and a distinctive R group. This is referred to as the side chain because it's going to come off the side of your polymer chain. Okay? Now, anybody remember what alpha meant? So when we talked about functional groups in organic chemistry, alpha was always that first carbon after the functional group, right? That's why that's referred to as an alpha amino acid, an alpha carbon. It's because it has the functional groups attached, the carboxylic acid, the amino group, which is an amine, right? Nitrogen. And a hydrogen atom, which is not a functional group, but has one. And a distinctive R group. By the way, how many different things is that? How many deals are here? 4. And so 4 different things means it has what? Chirality. Yeah. So these are basically always chiral, okay? There's one that is not chiral, but you get the idea. Okay, so again, chirality, right? Amino acids are going to be chiral. Almost all of them are chiral except for one. These are going to have 4 different groups bonded to that alpha carbon atom. That's the alpha carbon atom. This is the R group, the hydrogen, the amine, and the carboxylic acid there. Notice that the amine has been protonated. The carboxylic acid has lost its proton, hasn't it? Because we have a base and an acid on the same molecule. Okay? So they are always going to exist with 2 different mirror images. These are called the L isomer and the D isomer. If you ever go into a health food shop and try to get amino acid supplements or protein supplements, they'll give you several different options. One will be the racemic mixture, and one will be a purified only one of these isomers mixture. Okay? That's because you can only use one of the isomers. The racemic mixture means they have half of its junk, and you end up pooping it out. Okay? The proteins are only going to contain the L isomers, this one right here. The D isomer is essentially waste material for the most part, though there is a few things that can happen. Yeah. Remember the DL isomers when we talked about chirality last semester? It's that. It's the directionality. Yeah. So see how the hydrogens are in the back? It's the right or left depending on priority. Okay? Yeah. And that's just straight up organic chemistry terminology. That's why you need Organic 1. Congratulations. Your last semester of suffering has been utilized. Okay. So a neutral pH free amino acids will exist as dipolar ions. These are called Zwitter ions. Anybody know where Zwitter comes from? Okay. I'm going to tell you that most organic chemistry things are actually German. Okay? And that's because organic chemistry's big blow-up moment where it started being developed was Germany. And in fact, that's one of the reasons Blitzkrieg warfare ever even happened, is because Germans would take speed when they were doing Blitzkrieg warfare. Yeah. That's how they could go 3 days, march 3 days straight. Yeah. Anyways, organic chemistry. Okay, so right here, you notice that we have a base and an acid on the same molecule. That is the amino group, which technically is an amino salt at this point, but whatever, we're biochemistry, we don't have to be perfect. And a carboxyl group, which is the COO minus right there. So notice that because this is an acid, it deprotonates at a fairly low pH, around 2 to 3, okay? And this is a base, and it has a pK around 9.7-ish, somewhere in there, a bit lower than 10. That we usually end up with a Zwitter ionic form, and almost all of it will end up in the Zwitter ionic form at physiological pH. Okay? It's very unusual not to have the Zwitter ion. Zwitter meaning two-handed, so you have two ions. Okay? Notice that the majority of the amino acids actually are neutral at physiological pH. Wait a second, I just said it has two charges. Dr. Clausen, what the hell are you talking about? What am I talking about? If something has a positive charge and a negative charge, what is the overall charge? Neutral. So most of them do end up neutral, but that's because the R group can be charged if it's certain ones. Yeah, we'll see. We'll talk about that. So the 20 amino acids, these contain unique side chains that vary in size, shape, charge, hydrogen bonding capacity, hydrophobic character, chemical reactivity. These can be placed into one of four groups, and your book has a nice table that you're going to want to write out in card form and study as groupings. So you have these in the back of your head. If you're taking any form of testing whatsoever towards the medical biochemistry fields, memorize these damn things perfectly with charge, everything, okay? Because you are going to be asked many questions about this on whatever standardized test. It is the question set because they're really fucking easy to write, right? Okay. So again, that's hydrophobic, polar, positively charged, and negatively charged. Does anybody have any questions about those groups? No, they make a sense, right? Yeah. So is the side chain have a positive charge on it? In which case, at physiological pH, positively charged ones, right? Does it have a negative charge on it at physiological pH? Then it's negative charge overall, right? Is it polar? Polar meaning it can hydrogen bond, right? Is it hydrophobic? Meaning is water-fearing, right? And so would associate in hydrophobic structures, okay? They're going to have 3-letter abbreviations and 1-letter symbols. On your flip cards, you're going to want to add those and start doing them. You should go ahead and make those flip cards yesterday. But excluding yesterday, today. Okay? Okay. This right here is the hydrophobic amino acids. If you do not want to try to write these out yourself, which I do not blame you, you can take my figure, copy this figure off of it, and use a photo editing to and create your little flip card thing, either app or actual flip cards. So this right here is glycine, 3-letter gly, G, okay, for 1 letter. And notice the hydrogen here, okay? This one is somewhat unique. Can anybody tell me why this one would be unique? How many things around the carbon are there that are different? 3. This one's not chiral, okay? That's why I kept having that, like, exclusion. Okay, I'm actually just going to talk about the unique ones here, but no, these are the hydrophobic ones, meaning that the side chains here are not water-loving, right? They're water-fearing. They don't dissolve in water. They don't hydrogen bond. They won't associate in ionic salt bridges for the most part. None of that stuff, okay? Anyways, the kind of unique ones, proline is probably the most unique one. See how it wraps back in on itself and it actually bonds to the nitrogen, okay? So it has a very, very unique structure, and it has a very unique structural indications, okay, results of that structure. Because it, it has more rigidity than a normal one, doesn't it? Okay. The tryptophan and phenylalanine, these are very big hydrophobic groups, and so they can have actually very strong van der Waals forces. If you see something like that, it has some strong van der Waals forces pulling stuff together, okay? Like if you see a chain of those, several of those, it's going to associate to something. They're going to stick, okay? Big groups. Notice the methionine, methyl, met, and then thionine. It's actually just the organic name for that. And that has all the chemical properties of that methylphthalate groups. So if you're ever curious, yes, it does. Any questions about that so far? The charged ones, can I have a positively charged or negatively charged one that's a hydrophobic one? I guess I should have just said positively charged. Any positively charged or negatively charged ones here that has an overall charge? Not really, right? Not these physiological pHs. See that? But this one actually can become charged. But it takes some energy. It's not easy to do. Okay? But no, normally they aren't going to be charged because they don't have lone pairs, which is why they don't hydrogen bond, right? Yeah. So these are going to have non-polar side chains of different shapes and sizes. The shape and size is actually very meaningful, by the way. That determines what will work for binding different things. Okay. The lack of the ability to interact well with polar substances such as water is an incredibly important factor in these. It enables them to pack together to form compact structures. Remember how we talked about a lot of the driving force of creating these structures and shapes that we see is actually the hydrophobic interactions, right? That stacking, okay? And so these are the protein's way of causing hydrophobic interactions and stacking, aren't they? So, very important. The glycine is actually the simplest amino acids. It's achiral, and it's sort of the baseline. Doesn't have a lot of volume. It doesn't have anything, really. Okay? It can twist around and change shape very easily. And it's hydrophobic, non-polar, but it's such a small group that doesn't really matter that much. Tryptophan is the bulkiest hydrophobic amino acid. This actually has called an indole group on its side chain. That's that group right there, the red one. And it's more conformationally restricted than any other amino acid because it has this huge group that, I mean, you can't fit it anywhere. It means you can't bend in most of the directions possible, right? Because it's this huge, bulky group. And so it has a very large effect anytime you see that. It's kind of an interesting one. These are the polar amino acids, though some of them are not as polar as you probably are thinking. Okay? But some of them are incredibly polar. So these right here are some the amino acids that are polar. Here's serine. Again, make sure you memorize those 3-letter and 1-letter codes. I'm not going to concentrate on testing you a whole lot on that, but if you get out into the med school, PA school version of this class, and you don't have those memorized, you're going to really, really regret it. Okay? So start now, so when you get into later things, it won't be an issue. And also, I will have some testing on it, and some problems will use the 3-letter, 1-letter code. Okay? Though again, you don't fail a class because of one problem. So, let's see, tyrosine here. This is when I was talking about some of them are not as polar as you might think. This is that example, right? And this one right here, histidine. You guys probably all recognize histidine. Yeah, look at that. That's what that looks like. That's what that was this whole time. Yeah, that is one of the polar ones. It has a reasonable amount of polarity, though it is fairly constrained in its shaping. Cysteine is probably the most interesting and meaningful side chain on here, because anytime you hear of, anybody know what causes curl in your hair? Thiol bridges, disulfide bridges. And it's 2 cysteines. The sulfurs are bonding together on 2 different amino acids. And they don't even have to be on the same area. They can be pretty far apart. And that binding actually is what causes the curl in your hair. and a bunch of other stuff. But curl in your hair is the one you see. So these are going to have a hydroxyl group typically that makes the polar amino acids much more hydrophilic. You guys know, water-loving. Reactive than hydrophobic amino acids. Keep in mind these electronegative groups like alcohols, thiols, aminos, amides. These are things we talked about, a lot of reactions in general in organic chemistry, right? And then you're probably talking even more about those in organic too, aren't you? And so these are things that show up in reactions a lot, right? And these are spaces where you look and see, hey, if I'm going to have a reaction, where am I going to have it? There, right? Also keep in mind, for example, right here, see this little star? What does that star mean? How many different things are bonded to that? It's chiral, yeah. That's chiral, isn't it? There's a hydrogen you're not seeing, right? So there's 4 things. Okay, so cysteine, this is going to contain a sulfhydryl, which is another naming for thiol, by the way, but nobody ever uses, which is an SH group. This pairs with the SH groups, can come together to form the disulfide bonds, which I had mentioned. These are incredibly important when we talk about protein stabilization, creating structures of proteins, and large bulk structures that you see and exhibited in your body, like around you. Yeah. Histidine, this is often found in enzyme active sites. This indole ring combine and release protons during enzymatic reactions because it has a very interesting interplay with the conjugation of this ring. Notice this ring is a conjugated ring with one lone pair out of the plane of that conjugation and one lone pair that's in, right? And so you can have very interesting effects with gaining and releasing protons with this ring, okay? And so if you look for an active site on many enzyme reactions, you're going to see histidine involved, okay? The cysteine's not unusual to see, especially on like binding sites for things like irons and stuff, but the histidines are very, very common in reactive sites. This is the histidine right here. This is one with the that's been protonated. And so this has a pKa value that's near 6. This is pretty close to the physiological pH, which means that you do have a small population of the histidines that are going to be protonated and positively charged, okay? Though the majority of your population will be this form, which is the neutral form. What that also means is that you can protonate and deprotonate them in physiological pH conditions, okay? And so you can move protons on and off during reactions with relatively low energy expenditures, relatively low activation energies. Okay, so these are the positively charged amino acids. These are the ones that the majority of the form will be positively overall charged in physiological pH, right? And so this is lysine and arginine. Notice it's arg like a pirate. It even kind of looks like a hook. Ooh, ah. By the way, if you learn well with songs or mnemonics, there are hundreds of them available on whatever website you look. It is really drastic. Any beat, song you want, okay? Okay. Anyways, those are positively charged. The positively charged is going to refer to likely charge of physiological pH, of course, because many of the ones we looked at, for example, the histidine can be positively charged under certain conditions, right? So this is the complete positive charges render these amino acids highly hydrophilic because ions bond really well to water, right? And so these are highly hydrophilic. The arginine is going to contain a guanidinium group. That's the guanidinium group right there, by the way. It has some very interesting chemistry because of this. See how it has like this resonance structure going on, where it has half bonds everywhere? Yeah. So that would be a resonance structure, right, if you were going to write that down. Okay? And so it shares the charge among those nitrogen atoms, meaning it's surprisingly stable like that. Okay? Very interesting group. Again, that's a guanidinium group, in case you're curious. Okay, so these right here are going to be the negatively charged amino acids. Does anybody know what sort of group that is? It's like carboxylate, isn't it? Okay, notice all the negative amino acids are carboxylate groups. Okay, as in carboxylic acid lost its proton, as in the exact same thing down here. Right? Okay, and the only difference between them is one has one carbon here, one has two carbons. And then the functional group. Okay? All right, so negative charge again, physiological pH, negative charge. That's what that grouping means. The aspartic acid and glutamic acid, these are often called aspartate and glutamate. This emphasizes that the side chain usually lack the proton and are almost never present in the acidic form at physiological pH. Okay? And so again, this H right here actually refers to the same thing as carboxylate. That's where that comes from, doesn't it? Yeah. Carboxylic acid goes to carboxylate when it loses its proton. And that's why the naming was like that. By the way, if it can help you to learn these names by knowing that these names are actually somewhat based on, kind of as best as they could, the organic chemistry of it. So the ines, these refer to positively charged amines right here. Ine, amine. And so on here. Threonine, 3 different groups. See that? I'm going to not go into a huge amount of detail because it takes a lot of time and I don't think it really helps that much. Because my one that works for me doesn't work for you. And there are really fun videos and I'm not that fun. Take the chance. Go on to whichever streaming service you use. Okay. So here, right here, we have the different amino acids with readily ionizable side chains. These would be the typical pKa that you would see that interchange occur. And remember that at pH equals pKa, you'll get 50% of each species, right? The protonated versus deprotonated. Okay? And these are the different functional groups that we've seen. Anybody have any questions about that? Nope. Keep in mind, we are back to a water-based system. So that organic chemistry of like pH 15, that's a pretty good acid. It's not a good acid, is it? That's pH 15, pKa 15, that's a base. It's a really strong base, right? Okay. How's timing? Okay. You guys do this and we're gone. Fantastic. Okay, we'll start here next time. Wow, I'm actually doing okay on time, aren't I? I'll get pretty close to finished. So I need to take a medium-sized knife